This is Professor Doherty, and today we're going to be working on Lab 5, Diffusion across a membrane. Before we get started, let's review some important terminology for this lab. Diffusion is the movement of solutes from areas of high concentration to areas of low concentration. Recall that this requires no input of energy and does not require any form of active transport. Next is osmosis. Osmosis involves the movement of water molecules, and recall that we can use the phrase water chases solutes to illustrate the fact that water molecules are attracted to areas of high solute concentrations. Next is semi-permeable membranes. You should understand by now that semi-permeable membranes are non-living membranes that allow molecules through based on the size of the holes in the membrane in comparison to the size of the molecules attempting to get through those holes. We then have words that describe the molecules attempting to cross the membrane. Penetrating solutes are molecules that are small enough to pass through the holes in the semi-permeable membrane. Non-penetrating solutes are molecules that are too large to pass through the holes in the semi-permeable membrane. Now I'll draw a quick demonstration of how a semi-permeable membrane works. Imagine, if you will, two beakers adjoined by a connecting tube, and that tube is blocked with a semi-permeable membrane. The two beakers are filled with fluid, and a large molecule, non-penetrating solute, is added to the beaker on the left. This molecule is larger than the pore size in the membrane and therefore cannot pass over to the beaker on the right. Now imagine that we add a small, penetrating solute to the beaker on the left also. Because this molecule is small enough to penetrate through the holes of the membrane, it will eventually diffuse over to the beaker on the right until it comes into equilibrium. In this system that we have depicted here, the process of osmosis is also going to take place. Notice that we have more molecules in the beaker on the left than we do on the right, because the beaker on the left contains non-penetrating solutes. These extra solutes in the left beaker will attract water molecules from the beaker on the right, and subsequently we will see that the water level of the left beaker will rise while the water level of the beaker on the right will fall. This is due to the process of osmosis. This is why we say water chases solutes. Now that you have the basics down, let's take a look at today's lab. In today's lab, we will be running 3 tests for solutes that you need to understand, so I recommend that you write down these changes that I'm going to talk about here. The first is called a Lugol's test. This is an iodine-based solution with a chemical formula of I2Ki, and this molecule tests for starch. If this molecule is placed into a solution with no starch present, the solution will appear brown in color. So you should write that down. A negative Lugol's test for starch appears brown or light brown. If Lugol's is placed into a solution that contains starch, the solution will turn a dark purple color or maybe even a black color. Our second test is called a Biuret's test. This is a dye that changes color in the presence of protein. So Biuret's test is for protein. If no protein is present, then the solution will then appear blue. If protein is present, then the solution will appear violet in color. Finally, our third test will be a dipstick test for the presence of glucose. If you dip the stick into a solution that contains no glucose, then the pad on the end of the stick will remain the same color, which is typically a light bluish or greenish color. But if you dip the stick into a solution that contains glucose, then the pad will turn brown. Now let's discuss what today's lab looks like. Today's lab involves using 2 beakers, one of which will be used as a control that has water in it, and the other will be our experimental beaker. Here I am only depicting the experimental beaker, but we will be doing the same process to the control beaker, with the exception that we will only use water when we make the dialysis bag. To begin with, we will add 90 milliliters of water along with 10 milliliters of Lugol's solution, which if you recall is a test for starch, into the beaker. Recall that Lugol's will turn dark if it comes into contact with starch. We will then take a piece of dialysis tubing and form a bag out of it. by binding one end. We will then place 10 milliliters of dialysis solution into the dialysis bag. This solution contains 10% glucose, 0.5% starch, and 1.5% protein. You can find a listing of the dialysis solution contents at the bottom of page 2 of your handout. We will then seal the other end of the dialysis bag to make a closed bag. Remember that the dialysis bag is a semi-permeable membrane, meaning that the bag has microscopic holes in it that will allow molecules smaller than the pore size to pass through these holes. These would be called penetrating solutes. We then take the bag that's filled and we weigh it to determine its starting weight. We will then place the dialysis bag into the beaker containing the Lugol solution and then wait 75 minutes to allow any small molecules to penetrate through the dialysis bag and into the surrounding solution, or vice versa. After 75 minutes, we will then run our 3 tests for solutes to determine if any of the solutes have penetrated into or out of the bag. Our first test will be the Lugol's test for starch. Lugol's itself is a very small molecule. Since we know that there is starch in our dialysis bag, if the internal contents of the bag turn dark, this would indicate that Lugol's was able to penetrate the dialysis tubing. If the starch molecules are small enough to be able to exit through the pores of the dialysis tubing, we would then expect the solution in the beaker to turn a dark color. Our next test will be the Biuret's test for protein. One thing I'm not depicting here is that the Biuret's test will actually take place in test tubes, where we will place a small sample of the beaker water and a small sample of the dialysis bag fluid into test tubes and then add Biuret's to those individual samples. Here's what should happen to the solutions if they contain protein. If we mix the dialysis bag contents with Biuret's solution, we would expect the bag contents to turn violet, since we know that the bag contains protein. If the protein molecules from the dialysis bag are small enough to pass through the dialysis membrane, then we would expect the beaker water to turn violet when Biuret's is added to it as well. Recall that a blue color would be a negative test for protein. Our final solute test is a glucose dipstick test. For this test, we take a dipstick that has a pad on the end of it that is sensitive to glucose and place the dipstick into solutions from the dialysis bag. We know that this solution has glucose in it, so we should get a positive dipstick test, which means the pad on the end of the strip should turn brown. What we don't know is whether glucose moved from the bag into the beaker through the dialysis tubing. We can check for this by dipping a glucose test stick into the beaker water. If the pad on the stick turns brown, then glucose has moved from the bag into the beaker fluid. Then we have one final test, which is the test for osmosis. When the 75-minute wait time is over, we will remove the dialysis bag from the beaker and weigh it. We will record this weight on the table on page 3 of your lab handout and compare the end weight of our bag to the starting weight of the bag. If the weight of the bag has increased, this would indicate that osmosis has occurred with water moving into the bag. If the weight of the bag has decreased, then this would indicate that water moved out of the bag. If the weight of the bag stayed about the same, then no osmosis has occurred. Now that you have an understanding of the procedures for this lab, let's go ahead and get the lab started. Here I have prepared 2 beakers, each containing 90 ml of water and 10 ml of glucose solution. The beaker on the left will be for our experimental dialysis solution, and the beaker on the right will be for our controlled dialysis bag, which will contain only water in the bag. This is step 8 on page 3 of your lab handout. Next, I prepared our controlled dialysis bag. This bag contains 10 ml of water with no solutes in it. I will then weigh this bag, and you can see that the bag weight is 9.99 grams. You should record this number on page 3, table 1, under weight at time 0, in the negative control equals water row. We also should note here that the bag contents are clear. You should record the bag content color on table 1 of page 3 in your lab handout under initial color of bag contents for the negative control of water. I then take our control bag and place it into the control beaker. Next, I make a dialysis bag and fill it with 10 mL of dialysis solution, which you can see here. Recall that this solution contains glucose, starch, and proteins. You can also see the color of the bag here. You should note that the contents of the bag are white in color, and you should note this on table 1 on page 3 in your lab handout. I then dry the outside of the bag and weigh it. The weight of this bag is 10.59 grams. You should record this as your starting weight for the experimental bag in table 1 on page 3 of your lab handout. I then place the experimental bag into the experimental beaker on the left. After 75 minutes, I retrieve the control bag from the control beaker and blot dry the bag to remove any extraneous water. Note that the contents of the bag have changed in color. The bag contents are now light brown when initially they were clear. This indicates to us that the Lugol solution from the beaker has penetrated into the bag. You should record this color, light brown, as the final control bag color on page 2 in the table. After thoroughly drying the bag, we then weigh the bag and record the final weight. Here you can see the final bag weight is 9.91 grams. So the bag lost 0.08 grams in 75 minutes. This is a very small amount of weight, which is within our margin of error for weighing. So we would consider the bag start and end weight to be nearly identical. This indicates that no osmosis has occurred into or out of the control bag, which is what we expected. I'm now jumping around in your lab a bit here. I'm now doing the glucose dipstick test into the control beaker water. This is found on page 5 of your lab manual. After dipping the stick and waiting 30 seconds, I'll then compare the color of the dipstick pad to the colors on the side of the dipstick container. These are the standards. You can see that there was a very slight change in color to the pad. This is probably due to the fact that I'm using household water and not sterile deionized water. So we would record this as a negative result for glucose, since that stick did not turn a deep color of brown. You should record this on page 5, table 2, under Results of Glucose Dipstick in the row entitled Instructor's Negative Control Beaker. Our next step is to remove the experimental bag from the experimental beaker. Note the color within the bag. The color within the bag for a positive Lugol's test should be dark purple. We see only a brown color here, which is a negative Lugol's test for starch. What this indicates to us is that either the starch within the bag broke down before we ran the experiment, or someone forgot to add starch to the dialysis solution. The inset picture here shows what we should be seeing, which is a very dark purple bag. For our purposes, we're going to pretend like the bag has starch in it. So we're going to record the color as dark purple. Now I will tell you that starch is too large of a molecule to move through the pores of the bag, which is why we see a brown color still within the solution of the beaker, indicating that no starch has moved through the bag walls. I then thoroughly pad dry the experimental bag and then weigh the bag. You should record the final weight of the experimental bag in table 1 on page 3 of your lab handout as 12.86 grams. You should also note the color of the bag in this table. I then place the experimental bag into its own small beaker and I pour the bag contents into the beaker for easier access to the bag solution.
I then take 2 glucose dipsticks and dip them into the experimental beaker water and the experimental dialysis bag solution and wait 30 seconds before checking the strips against the color standards on the side of the glucose stick bottle. You can see that both of these dipsticks turned a moderate shade of brown, which is a positive indicator for glucose. This indicates that glucose was a small enough molecule that it was able to move through the dialysis bag pores and into the beaker fluid. You should now record the 2 positive glucose test results for the experimental bag and beaker into Table 2 on page 5. Our final test is to find whether or not the protein from our experimental bag was able to pass through the dialysis bag pores and into the surrounding beaker water. To do this, we place 5 milliliters of the experimental bag contents into a test tube and label it EB, which is on the left in this picture here. Then we place 5 milliliters of the experimental beaker solution into a test tube and label it ED, which is our middle tube here. And finally, we place 5 milliliters of the control beaker water into a test tube labeled CD, which is the test tube on the right. Next, I will place 3 milliliters of Biuret's reagent into each tube and observe the reactions. I'll start with the control. We take the control test tube on the right, and you can see that the tube contents turn blue when I add Biuret. This is a negative indicator for protein, which is as we would expect because we know that our control only had water in it. Next, we place Biuret solution into the solution from the experimental beaker. And again, we see a blue color, indicating that no protein is present. Finally, we place the Biurets into the solution from the experimental bag on the left-hand side here, and we can see we get a distinct violet color, which is a positive indicator for proteins. You should record these results in Table 2 on page 5 under Results of Biuret Test for Proteins. Make a note to yourself that these results indicate that protein was too large of a molecule to move through the dialysis bag pores from the inside of the dialysis bag out into the surrounding solution.
This concludes the experimentation portion of Lab 5. You should now be able to answer all the questions throughout the lab handout. I would like you to note also that on page 2, in the bottom section of the page, that the molecular weights of all of the biomolecules that we've been working with today are listed. This should help you with the remaining questions in the lab handout. Thank you.